So before I start my talk, I also need to give out the Presidential Award for the American Society of Naturalists, which is the choice of the president that year of the best, there's no such thing as a best paper, but the paper that appealed to the president the most, that was published in the year preceding or during their tenure as president. And so this year I chose the paper Evolutionary Assembly of Communities of Butterfly Mimicry Rings by Janavi Joshi, Anupama Prakash, and Krishnamay Kunte. And I chose this paper because of its synthesis of a lot of natural history information to give us new insights into mimicry. These authors looked at island and mainland mimicry rings, the evolutionary origin and assembly of those rings, which was interesting. And my favorite part of this paper was that looking at Batesian mimicry in butterflies, they found that the ratio of models to mimics was maintained across mainlands and islands, even when the island population, the island diversity was much lower, which I thought was a very cool result. And so for this really insightful paper, an original paper, I chose this paper for the Presidential Award. So can you please come up and receive your award? Congratulations. Congratulations. So now, living in deathly fear of cicadas and music, I will continue. So the title of my talk reflects the dual nature of my talk, which is diversity and coexistence in close relatives, and reflections on 150-ish years, you'll get the ish later, of American naturalists. And but first I wanted to say I'm really honored to be here, very grateful to be part of this incredible meeting, which is amazing, and thank Ophélie and all the other organizers for the really great job they've been doing here. So when we go into natural communities, we're often struck by the diversity that's there, and that diversity arises from some combination of historical and current processes. So the historical processes are, how long has this piece of land been uplifted, and what is its geological history? What are the centers of origin of the clades that are inhabiting that area? More recently, historically, might be what kind of interactions and evolution have taken place that allow species to coexist. And then there's sort of the current ecology going on. How do these species not exclude each other from that habitat? So I watch other people who do exactly that. Coexistence in closest relatives, so thinking congener type relatives, provide us with, I think, unique insights into the processes that govern diversity, in that these species have very recently speciated, so we might expect them to be very similar, and if they coexist, these are actually two pairs of congeners at my field site, and you can see they look very similar. So and they're within dispersal distance, within gene exchange distance. How is it that these coexist? Okay. So we often think of coexistence in close relatives in the context of adaptive radiations, things like Galapagos finches or cichlids, where we have sort of sympatric speciation into new habitats, and sort of a focus on coexistence in sister species, and how did that arise. But I'm hoping to argue that there's a lot of interesting information about the assembly of communities and evolution if we consider congeners that are more distantly related than sisters. So if we think about what are the pathways that have to be satisfied for coexistence of any species, we might think that there's some allopatric speciation event, that if these species never come into secondary contact, well, then they'll never coexist. So, okay, they have to come into secondary contact. If at that contact they hybridize, they're also not going to coexist. They're going to collapse back into one species. So then, if they're reproductively isolated, then can these species coexist ecologically? So they're not excluding each other through either competition or higher order interactions or whatever mechanisms we know result in exclusion. And if they can't, they also won't coexist. So really we need to satisfy all of those criteria to get local coexistence. And for sympatric speciation, well, we bypass a lot of those intermediate steps, but there's still the coexist ecologically that's going to be critically important. So Darwin, standing on the shoulders of giants, in this case, maybe I'm holding him up, but has, of course, made comments about so much of everybody here has been quoting Darwin. Sadly, I'm no exception. But he wrote, as species of the same genus usually have much similarity in habits and constitution and always in structure, the struggle will generally be more severe between them if they come into competition with each other than between the species of distinct genera. So let's unpack this a little bit. As species of the same genus usually have much similarity in habits and constitution, then this might suggest that they like the same habitats and the same conditions, and we might expect them to coexist. But then we have the flip side, that the struggle will generally be more severe between them, in which case we might expect that one they should exclude the other and that they should not coexist. So a roadmap of my talk is going to be, are close relatives, 2 parts, are close relatives ecologically similar and do they co-occur? And what other processes might influence coexistence in reproductively isolated close relatives? And then the second part of my talk will be this sort of abrupt shift, and then reflections on the history of our society and also our future as naturalists. So in the first part, I'm going to attack these questions that Darwin posed. Do closely related species prefer the same habitats? Do they compete more strongly? And ultimately, do they coexist locally or not? And so this question has been addressed more broadly under an umbrella of questions. Does phylogenetic relatedness predict ecological similarity and community composition? And typically, this question has been addressed descriptively. So going out and observing, or observationally, observing communities and then looking at the patterns that occur. So if we have 3 species communities here, and there's some relationship, we can see that in these 3 species communities, these are actually close relatives, and that's true for each of these communities. And so if we look at the relationship between phylogenetic distance between species and the rates of their co-occurrence, we see that close relatives coexist a lot, and we have a number of examples from our colleagues. And again, these cannot be exhaustive reviews. I apologize for anyone I've left off these slides because there are so many people who have been working in many of the fields I'm going to be talking about. We can also see the other pattern where we have communities assembled by one member from each of these subclades where a coexistence between close relatives is very rare. And again, we have many studies that show this pattern as well. So my lab takes a different approach. We call it the Oprah approach, because we can take advantage of species being sessile and not being able to move, particularly plants. This is why I study plants. I love animals, but I study plants because I can torture them. And I can plant them into the niches of other species that vary in their relatedness to that planted species, and then I can interview them by asking them, you know, how do you grow and survive and generally perform with that relative? So to do that, we're working out at Bodega Bay Marine Reserve. This is actually a small reserve. This is a scale of 1 kilometer. Thanks to the San Andreas Fault, which runs through the reserve, we actually have this very large diversity of habitats where we have extensive sand dunes here, and then we have this coastal grassland we have some wetlands, we have this sort of rocky bluff. So we have a lot of quite different habitats in very short spatial distances that seeds can move around in, that pollinators can fly across, that plenty of opportunities for gene exchange and dispersal, ecological exchange, and this is work done with Brian Anaker. And what we used is this field transplant design where we took a focal species, in this case Plantago erecta, and then we planted it into other conspecific sites. So what I mean by these sites is where these species are very dense. So conspecific sites, congener sites, so here Plantago subnuda, confamilial sites, and distant relative sites, which are extra family sites. I want to point out that none of these congeners that we know so we don't have complete phylogenies for most of the groups, but for the groups where we had complete phylogenies, I can tell you none of them are sister. So we did this, there were 2 replicate sites per each one of these sites, types, and we had 15 species sets, and we actually did this for 2 years. And so we have a quite large experiment, and we only did these with native forbs. And while treatments were assigned with rank based on the species list, we then created a molecular phylogeny based on 4 genes so that we could measure the phylogenetic distance separating species using genetic distances. So I'm going to show a couple of sets of slides where we always have the phylogenetic distance separating the focal planted species from the resident here on the x-axis, and then some metric of performance here. So if we look at survival in relation to phylogenetic distance between these species, what we found is that we had similar survival to the family level. So the mean divergence was about 63 million years of confamilials, and this is about where we start to see declines or shifts in our slope in each of our experiments. And so we see a moderate decline in survival at distant relative sites and similar survival at closer relative sites. So to understand the role of biotic interactions, like do they compete more strongly, half of our replicates were weeded to remove the local neighbors. It's just a small amount of weeding, and then the others were left intact. And this is into the ambient community, so there were high density of the focal relative, but other species were left there. And then we looked at, I wanted to explain RII, well, I'll do that. So relative interaction intensity is our metric. A negative value represents competition. This represents sort of positive facilitation interactions. So if we see what we see here is that close relatives suppress each other more, and even if you remove the conspecifics, there's still a positive slope. So distant relatives actually often facilitate one another. So if we then look at how do species actually perform the biomass of the survivors in these sites, what we get is this curvilinear relationship where species do best at conspecific and distant relative sites and poorly or less well in congener and confamilial sites. And if we sort of multiply survival rate by performance, we basically get the same pattern, except this drops down a little bit to be even more even here. So these results would suggest that we would predict spatial repulsion of congeners and confamilials at the reserve. So then we went to ask whether or not our experiments could actually predict the patterns in this 200 plus native forb species community at Bodega. And first, because we don't have phylogenies and molecular data for all of these species, we first went back to our original data and said, okay, well, we originally assigned these through ranks. Are our results recapitulated if we consider ranks? And what we see is the same basic pattern of better survivorship in conspecific and distant relatives and lower survivorship in congener and confamilial sites. And so we could place out quadrats and look at well, what species are in these small, these are a quarter meter on a side, and what species do we find in there? So inside this plot, there are 20 species from 19 genera. And so we can calculate the probability of encountering a specific rank at these local, really local coexistence scale, and then also as a function of spatial distance separating quadrats. How likely are you to encounter, what is the relationship of when you start to encounter particular ranks? And what we found was that rates of co-occurrence with congeners was very low and was way less common than expected by chance, and this is what our data predicted. Although congeners, but not confamilials, were more spatially dispersed. So to address what Darwin said, congeners are more ecologically similar, they survive better in each other's sites, and they compete more strongly with each other, so there's the conflicting effects of ecological similarity. And the net result of that at Bodega was fine-scale spatial repulsion of congeners. But why are only congeners spatially repulsed if we see the same magnitude of effects for confamilials? And so, what other forces might drive habitat partitioning in close relatives? And I would argue, and a number of my colleagues have been arguing for a while, that reproductive interference may be a very important mechanism affecting local coexistence of congeners. So what is reproductive interference? It's actually an interspecific interaction between reproductively isolated species during mating or mating processes that reduces the fitness of at least one species. That's a little bit hard to understand. I'm just going to go through it. So basically, in both plants and animals, reproductive interference is strongest in congeners. And what you have are situations where one species attempts to mate with the other species, and there are never any hybrid offspring that are produced. But there is a certain fitness cost to this heterospecific mating attempt. So in this case, we have this Rana dalmatina that attempts to copulate with Rana temporaria, actually reduces the rates of egg fertilization of Rana temporaria and has significant fitness costs where they co-occur. So you might imagine that sessile species are going to experience this and actually have no opportunities to leave sites where heterospecific matings might be messing with their fitness. So opportunities for these species are if pollinators clog stigmas with heterospecific pollen, or if they export pollen to another species, then that's also a fitness cost. So we might expect this process to be especially important for sessile species like plants. And so we expect reproductive interference to be an important important when we have reproduction at the same time, species that look similar and that occupy the same or nearby habitats. And those criteria could easily be met by close relatives like congeners. So I talked to my colleague Sebastian Schreiber, who's upstairs, and I said to him, so we have all this Chesson coexistence theory where being rare is an advantage, because when a species is rare and invades a patch, then it's released from the resident, which is at high density, is busy conspecifically suppressing each other at great magnitude. The rare invader can come in because it's released from that competition, it's not experiencing that strongly, and can increase in density when rare. But what if when you're rare, you get more heterospecific mating attempts, which we know from in the literature in reproductive interference that being rare is sort of a key part of this disadvantage. So Sebastian and Masato Yamamichi put together these models where we have the same degree of niche overlap, the same ratio of fecundities that we would need in these Chessonian type coexistence models, and we can find that the rates of coexistence are greatly reduced even with 10% reproductive interference. And when we've looked at magnitudes of reproductive interference across a large number of taxa, it's often at least that. So this isn't just a hypothetical. There are a number of examples. This is one of my favorite examples from plants. This is work done by Ryan Briscoe Wallace and Maureen Stanton. These are two species of meadow foam, Limnanthes rosea and alba. And you can see from their ranges that they have very similar ranges. They live in vernal pools, and this is the study site where Ryan was working. And they're actually never in the same pool, even though some of these pools are only separated by 100 meters. So she began to explore the possible role of something like reproductive interference, which goes by so many different names in the literature: satirization for animals, competition via pollination is what they called it. But basically, she set out the arrays, and actually both species are in this array, so just to give you a sense of how similar they actually look. And then she created arrays where the frequency of one species was basically declined over the course of these arrays with the same number of individuals. And so then she can look at seeds per plant. And what she did was she set these out in pots so that there is no resource competition going on here. She just set them out in these little containers that she sunk into the ground, so they were growing, as if they were a little patch, but they were never exchanging any kind of interaction below ground. And what she finds is that as rosea becomes much more rare in these arrays, that it suffers an almost 50% decrease in seed set. And alba did not have as big of a decline, but it did have a non-significant decline as well. So in the absence of competition, we see this decline as a response to being rare. And they argue that reproductive interference may partially explain this fine-scale exclusion in Limnanthes. So I've just become more and more interested in this topic, and I thought, I have a lot of data on Streptanthus. Can historical approaches provide us any insights into the possible role of reproductive interference? So this is a group of mustards. They're called jewel flowers because they're really beautiful. They don't look like any other mustard that you've ever seen. Oh, shoot. So this is what they look like. And we have generated a complete phylogeny for the group. And so we can look at the evolution of traits. And in particular, we were interested in the evolution of floral attraction traits in relation to sympatry and allopatry in these close relatives. And the first part of this was not actually part of the original study, but is a key piece of information that we need, which is that Kyle Christie, one of my current students, as part of his thesis looking at the evolution of reproductive isolation across this clade, created many crosses that span phylogenetic distance between species to look at what are the mechanisms of reproductive isolation across the group. And all species are reproductively isolated. So this is good because we know that there's not going to be selection on floral traits due to hybridization. But what Kyle found was that more closely related species form inviable seed when they get heterospecific pollen from their congeners. So here, if we look at the effectiveness of prezygotic isolation, what we see is that when the genetic distance separating the cross species is low, when they're close relatives, a number of species are making this inviable seed, whereas as soon as they cross some threshold, we get no effect on seed set as a result of heterospecific pollen deposition. So more closely related species form inviable seed and may waste resources, whereas more diverged species make no seed with heterospecific pollen. All right, so let's look at range overlap as a function of the genetic distance separating these species. And what we find is, contrary to examples in animals, that more closely related species have greater range overlap. So this is the amount of range overlap, and again, the genetic distance separating species. And actually, this is a pattern that's not unique to this group, but in a survey that Brian Anaker did, we found this for every set of species, on average as a whole across the California floristic province. So less prezygotic isolation and more range overlap in close relatives means that there's opportunities for reproductive interference. So getting back to our original question, how do floral traits evolve? We measured flower size, and we also measured floral scent across 22 species in the clade. And so what we find is that close relatives have more similar flower size. So basically, this is flower size here, and we see that the more closely related species are, the more likely they are to share a similar flower size. For scent, we isolated 53 unique compounds, and there are 0 to 13 compounds per species. And in contrast to floral size, floral scent is highly diverged between close relatives and less diverged as phylogenetic distance decreases. So what is the rate of scent divergence, the evolutionary rate of scent divergence as a function of whether or not ranges overlap or don't? What we found was that there was a much more rapid rate of scent divergence when species are in sympatry. So much faster evolution in scent when species are in sympatry. So we were pretty excited about this. And so this reproductive character displacement in congeners may reduce reproductive interference in sympatric close relatives. And there are a number of other mechanisms that could result in that pattern in the interaction between sympatric congeners, but we think that this is also consistent with a role for reproductive interference. So when we think about the processes that are determining coexistence of species, we often think about resource competition. We think about higher order interactions, for example, predators allowing two species of prey to coexist, the role of environmental heterogeneity in promoting coexistence. We know there are, evolutionary processes like character displacement that allow coexistence, but especially when we're thinking about coexistence in congeners, we and many other people in this field at this point are arguing that reproductive interference might be worth a more careful look as another possible mechanism that's limiting this coexistence between otherwise very similar species that might share habitat preferences, that might almost act as ecological equivalents in some ways, competing strongly with one another, just as conspecifics do. So, one way forward might be to continue looking at these kinds of historical approaches, and I don't think we have the methods there yet to do this, but it would be super great if we could actually measure rates of evolution of resource acquisition traits versus reproductive traits in sympatry and allopatry and get a sense of, okay, well, which are evolving faster when we see sympatry. That might give us a sense of what are the relative importance of these forces when two closely related species come into contact. This thing is very twitchy. Okay, so here's the abrupt turn where I now want to focus some time on the 150-ish years of American Naturalists. I want to talk a little bit about our past and a bunch about our future. So first, the ish part comes from the fact that actually the journal, the American Naturalist, is older than the American Society of Naturalists. So it's actually we're in our 151st year of the American Naturalist. It originally had to be privately funded which was funded by George Peabody, and there were 4 initial editors, and it's the oldest continuously published scientific publication in North America, and this is where the Europeans get to laugh because old in North America is like, oh, for Europeans, but that's okay. The mission of the journal was really to appeal to a broad audience of farmers, gardeners, artisans. Its aim is practical and economic as well as theoretical. It was inclusive of many perspectives on nature. That's something that we retain today. And there's a really cool talk, it's posted on YouTube, that Dan Bolnick, the current editor-in-chief, gave about 150 years of the American Naturalist, so I urge you to go and look at it. There's a lot of cool information there. Darn. So, okay. So the American Society of Naturalists is only 135 years, but the founders of the American Society of Naturalists were sort of a subset of the people who founded the journal. So we're really very enmeshed, and we became quickly associated. And it originally started as sort of an Eastern Seaboard kind of association. It branched off from the Association for the Advancement of Science, AAAS, and the themes were, the naturalists were frustrated that at AAAS meetings there were hardly any time for talks on on nature, because they were sharing it with geologists and medicine and all these other fields that were combined. And so they splintered off, and originally they couldn't look as if they were competing with AAAS, so this was on the business of the naturalist, methods in mounting, cutting, staining, etc., to distinguish themselves. But really, the idea was to create a society where more people could talk about nature. And it was always a society open to diverse disciplines, not specialized on, a particular taxon. And it quickly moved to be a forum for natural history research. And it was, importantly, a nursery for a lot of other specialized societies. So the American Society of Zoologists, now SICB, basically they started meeting with ASN, they built critical mass, and then they split off. And the Geological Society of America, and there are a number of other societies that did this. So ASN was sort of a nursery for many of these other societies. It was interesting, the first society to restrict its members to professionals by its constitution, and this is going to have some profound implications I will get to in a minute. So they should be limited to instructors of natural histories, officers of museums, and people generally professionally engaged. The first meeting was attended by 124 men in 1883. And while they started out trying to be this elite Eastern Seaboard kind of society, they rapidly just became more broad. So limiting members to professionals generally excluded women, and that may have been the intent. I don't know. There were a lot of women entering science. The AAAS was getting 8 women per year. As members in 1870. They actually then instituted 2 different classes of membership, fellows and members. And so it may not have been purposely for that, but it definitely had the effect of reducing the number of women as members in the society. Emily Gregory was a botanist, and she was the first woman to gain a foreign PhD in the US. So she got a PhD from the University of Zurich. And Ida Keller, shortly after her, followed in her footsteps exactly. And they were admitted as the first members of the ASN 3 years after it was founded. And I should just note that there are a number of other women with PhDs in the US at the time who were not members. So this foreign PhD gave them a certain panache. But even as members, they were often excluded from key functions. This is the 1907 dinner banquet of the ASN, the meeting banquet. And, I have looked through as many faces as I can possibly discern here, and I have been unable to locate a woman in this dinner. And it was wasn't written rule, but it was sort of understood that banquets were for men only, and this is where, a lot of networking happens. And the other thing that happened at a lot of these meetings were smokers. And smokers were where the guys get together and smoke. And because at that time, proper women didn't smoke, if you schedule key agenda items during a smoker, that's another great way of keeping women out of that important networking and decision-making process. So, we've come a long way. And if we look at the American Society of Naturalists now, we still are including a lot of diverse disciplines as in our membership and our publications, a continued tradition of excellence, and we're much more gender diverse. We don't have demographics, but we're in the process of getting demographics for our society. But just from looking at first names, we estimate approximately half of our membership are women now. So this is then, these are our meetings now, and that's really awesome. This is actually not an ASN meeting or even this meeting. It is the British Ecological Society, but it's okay. Look around the room. You can see that it's representative of what is in the room right now. And I really love this. I took part in the networking lunch yesterday where junior researchers meet senior researchers and talk about mentoring in their fields and advice. And I got this as the algorithm for creating groups, which was a clustering method to form 25 thematic clusters based on keywords supplied by the participants that were then randomly sampled for junior and senior members. And so, wow, this is cool. This is like networking opportunities that are randomized. We have really come a long way. So, look around the room again, because I still think we have more places to go. So let's think about the future, and not just the future of our societies, but the future of naturalists. What are projected global demographics? What are the demographics of green STEM? That's science, technology, engineering, and mathematics, and amateur naturalists. And what does this mean for the future of nature and naturalists? So by 2050, almost 70% of the world is going to live in urban environments. And we know that future populations are also going to be more diverse. These are just projections for the US population in 2060. And we also know that around the world, urban populations are more diverse than rural populations. So how do these global demographic trends mesh with STEM demographics? So when we look at diversity in STEM, we see that it's way less diverse than our global populations. And that if we look at green STEM, and it's hard to get these data. I actually got these from the National Science Foundation. This is graduate students in ecology, entomology, genetics, and zoology. This was about the closest I could get to what we are here. And you can see that it's way less diverse than our current populations. Okay, well, not all naturalists are professional. What about amateur naturalists like botanists and birders? We can define naturalist as pretty much anyone who is inexhaustively fascinated by biological diversity. Thank you, Doug Fatuma, for giving me beautiful words to describe that. And we know that, for example, many amateur naturalists are citizen scientists. So if we look at the demographics of citizen scientists, we see that African Americans, Latinas, Native Americans in the US participate less than other groups in citizen science, and that we generally have more affluent participants than the population as a whole. Birders are a huge source of naturalists all over the world, and there's very little information on the demographics of birders, but there was a survey done in the US, and you can see that birders are also way less diverse than the population as a whole. And then the sort of finishing touches are that urban dwellers are less likely to be naturalists. If we look at the participation rates, we see that people that live in rural areas are way more likely to be naturalists than people that live in cities. So we have a challenge, and that is, how do we meaningfully connect urban populations to nature? Because the demographics are telling us that, if we can't do that, we risk becoming both dinosaurs, but also the fate of dinosaurs, really. It may be the fate of naturalists in the future. So one thing to think about is that urban settings have nature, even if there's not a lot of economic resources to go out and see nature the way many of us love to see nature. There's still a lot of nature in urban settings. This is actually, I thought, an interesting futuristic view of an urban forest. And we know that even small urban green spaces can play key roles. So Access to small urban green spaces actually have been shown to have really positive effects on low-income neighborhoods. This is a really cool study that just came out. They assigned 540 vacant urban lots to no treatment, trash cleanup, removing weeds, and planting with grass and trees. And when we did that, they found less depression in neighboring people, better health metrics, and reduced crime. So this is great health-wise, but it also builds a foundation for urban naturalists just to have these green spaces that are accessible. Because we know that once exposed, some subset of us out there are going to get hooked on nature. And actually, I can say this because I grew up in the cement jungle of Manhattan. And people always ask me, how did you become an ecologist? And it's because I spent a lot of time in Central Park. I spent a lot of time at this amazing place, the American Museum of Natural History and zoos. And these are great places and accessible places to learn and get exposed to nature. And there are also a number of people who are actively working. Akima Price, is spending time in low-income neighborhoods and talking about the benefits of green and the nature and what it can do to serve low-income communities. City Blossom plants gardens. There's David Lindo, who is this manic urban birder in London, who does bird tours within London and surveyed the populace and found out that they picked the robin as their favorite bird. He's also a photographer and took that picture. And I'm not saying that we should change the missions of our societies, but our societies can participate in exposing urban populations to nature. And a model of this is the SEEDS program of the Ecological Society of America. It's now over 20 years old. And the focus was bringing urban and diverse students into nature and research experiences. And 20 years down there's chapters all over the US now after 20 years. And at this point, there's actually a sizable number of individuals that have gotten their PhDs after participating in this program. So exposure of urban populations to nature in many forms, I feel will be essential in creating the next generation of naturalists, because the demographics are telling us that these are the people who are going to be increasingly part of our populations, that they are going to be voting on putting resources into nature, and that we need to spend time making an effort, making nature something that is accessible to everyone. So this is then, this is now, and I hope that this is soon, that our audiences will reflect the populations in which we are embedded. And with that, I'm going to say thank you. Thank you to all of my colleagues who helped collect the data I talked about today, many generations of my labbies, and my colleagues at Davis who are great for joint lab meetings and discussion, my funding sources, and of course, this meeting and the organizers here. So thank you very much.